Good morning, everyone. That was a great introduction by Dr. Waters. It's going to make my job much easier. So my name is Nick Kovac. I'm a bioinformatics PhD student at the Georgia Institute of Technology. A bioinformaticist is someone who looks at biology using computers and statistics. So my advisor is Dr. Lauren Williams, and my presentation today is on the evolution of protein folding. So to begin with, like Dr. Waters was saying, the ribosome takes nucleic acid, which encodes information, and turns it into protein, which is function. So, that's the ribosome right there, and it's taking the RNA that comes from DNA, and it turns it into protein. And the ribosome itself is a large macromolecular structure of RNA and protein, and it does its function. So when you look at these phylogenetic trees that relate bacteria to archaea and eukaryotes, and you look at like all animals are distantly related to fungi and plants, that's by looking at the sequence of the ribosome on the DNA. So the ribosome encodes species relationship, and which is why we kind of say that the world started with the RNA world hypothesis. When you take the ribosome and you look at the size of it, and you look at the size of it within the organisms on the tree of life, you can see that prokaryotes have really small ribosomes that I have here represented by really small little circles, and that simple eukaryotes have larger ribosomes, metazoans, animals, have even larger, and then mammalian ribosomes are the largest found in nature. And what's very interesting is that these mammalian ribosomes contain the ribosomes of these animals. And the ribosomes of these animals contain within them the ribosomes of these yeasts, or simple eukaryotes. And these simple eukaryotes, the ribosomes of those, contain the ribosomes of prokaryotes. And so when you look at that, of the ribosomes of extant life, and look at the way that they've evolved since the last universal common ancestor, 3.5 or 3.8 billion years ago, and we just use the same mechanisms that we hypothesized that they've been growing since then. Luke is here, extant life is here, sorry about that. We can see exact same mechanisms that the ribosome, that relates to ribosome of all species in extant life today. If we use that, we can just deconstruct the ribosome and go back in time and see where life started with just the very first piece of RNA. And these very first pieces of RNA are the catalytic centers of the ribosome. And so that's where proteins are formed, right? At these little pieces of RNA. And so what we hypothesize is that the ribosome very early on in the beginning of life was making small little pieces of proteins, and as more pieces of RNA kind of stuck onto the ribosome, its ability to synthesize proteins of increasing complexity just started happening. And so we eventually ended up with a very functional ribosome that can encode, that can make very nice and complex proteins needed for life today. So I've been talking about the RNA of the ribosome so far, and I want to talk about the proteins of the ribosome. So when you look at the prokaryotic ribosome, there are 29 ribosomal proteins in just the large subunit. I'll be talking about just the large subunit today. This also applies to the small subunit. So there's 29 ribosomal proteins of the E. coli ribosome. And what's very interesting about these proteins is that a lot of them have these globular, like spherical domains that sit on the exterior of the ribosome. And those look like proteins that we find in life today. But they have these really weird segments that we just don't really find anywhere else. These tail segments, and they penetrate deep down into the core of the ribosome, close to where peptide bonds are actually made, where proteins are actually synthesized by the ribosome. And so looking at these proteins and what they contact of the ribosomal RNA, so at the very beginning you saw that I had one piece of RNA, and then as it was growing throughout the evolution of life, we can see that more RNA was conglomerating onto it. And if we look at the proteins of the ribosome and which pieces of RNA they contact, we can see a timeline of ribosomal evolution, or in just ribosomal protein evolution, and just protein evolution for that matter. So at the very beginning, we only had small little peptides that were sticking on there, but as the ribosome grew, the proteins that could stick onto it were growing as well. And I'm going to look at the structure of these proteins and see how it's changed since the origin of life. Okay, so a lot of these proteins are very positively charged because RNA is very negatively charged. So we see a lot of arginine and lysine, and hypothesize that we know that they kind of just hold the RNA together. They kind of just act as glue. So they're needed for the ribosome. Okay, and then so my background is in biochemistry, so I want to give you a brief background on what a popular plot that biochemists use called the Ramachandran plot. And so biochemists really like proteins and like protein structure. And so in proteins that we find today, we see a lot of protein, like looking at the angles that the amino acids in the protein form with each other. They typically, just the way they just fold into the protein, we typically find them in these centers right here in the red. And so we see this alpha helix, which is very common in proteins today, and that's usually just looking at the angles of those bonds. Usually find them right in this red region. And we also see a lot of beta sheets. That's another common secondary structure of proteins. And that usually falls within this red region. And then all this like white area over here, we don't find too many amino acids in there, but that's kind of unstructured. They usually don't like hydrogen bond with each other. You can't, it's just, they don't really form any like functional catalytic parts typically. Okay, so looking at the secondary structure of ribosomal proteins. So in the very beginning, you could see that I had this little peptide earlier on 2 slides ago. That was like composed of, I think, 12 amino acids, and approximately 1/3 of them fell within these red regions. And then when we look at the next little phase of evolution, a few more of the amino acids are starting to fall into these red regions. And as we keep on going through, we can see that more and more of these amino acids, the bonds between them, were falling into these red regions. So when the ribosome started producing proteins, they weren't very structured, but as the ribosome evolved, so did its ability to synthesize proteins of increasing complexity. And now they have these structures that we find in life, and many of them not within the ribosome, but these structures are often catalytic. Okay, so what about like hydrogen bonds within the ribosomal proteins? So I brought up earlier beta sheets and alpha helices, and they have a lot of hydrogen bonds. And so when I look at the phases of ribosomal evolution from the origin of life until last universal common ancestor right over here, we can see that the hydrogen bonds per residue are increasing. So these proteins are just folding more and more compact, and they're just looking more and more like proteins that we find in extant life today. And then when we look at the surface area of them by just a computational method of just pretty much just rolling a ball around the ribosomal proteins, we can see that their surface area is going down, and we just see that they're just folding more compactly, and that they're just looking more and more like proteins we find today. So I believe that is what I have for today. So my name is Nicholas Kovac, and I work in Dr. Lauren Williams' lab. And I really like to thank Dr. Anton Petrov and Dr. Chad Bernier, who have been my mentors. And I would just like to thank AgRadCon in Georgia Tech and the IBB at Georgia Tech for providing my funding.
Hi, I was wondering do you know what the minimum number of residues, like a polypeptide or I guess a short protein, if you define it that way, would have to like start exhibiting some type of defined structure?
There was recently a paper where just through like just directed evolution, I think it was like 12 amino acids, they could make a functional protein, when there are about 20 amino acids found today in all of life. And those 12, a lot of them, I think almost all of them, were actually the same amino acids found in the Miller-Urey experiment, which we hypothesized those are the very first amino acids of life.
I think you might have just answered my question, but I was going to ask, how did you determine the sequence of proteins for your early proteins?
Well, I was just looking at the crystal structures available of the ribosome today. So we have crystal structures of the E. coli ribosome, the yeast, Drosophila, human, and a few other species. And I was just taking the sequences found within those proteins of those ribosome crystal structures. Mm-hmm.
Yeah, thanks. Great talk. I find it interesting, this idea that species complexity is related to the size of the ribosome. Is that possibly because we have a lack of data on ribosomes that are out there yet? Or has this been shown across the board so far?
Mm-hmm. I mean, I'm looking at crystal structures, and I mean, there's just not that many of them available just yet. I mean, the very first one was like, we crystallized ribosome like in the early 2000s, so there's not that many of them, but we can look at the genes that encode for these ribosomes in species, fairly easily. We've been doing it for quite a while. And so that's to my knowledge, I think these mammalian ones are the largest we've seen.
Interesting. Mm-hmm. Because I know for a long time we thought that genome-wise that we humans would be the best. We'd have the most genes of anything, and that's not the case. And we've now found out that complexity in our little viewpoint of complexity isn't really related to genome size. And so I was just wondering if that would be the case with ribosomes once we get more data as well.
Yeah. Mm-hmm. Well, what's interesting is that a lot of that ribosome work was based on a paper we had, and we put it on. Someone picked it up on Reddit, and people were kind of getting really up in arms about big discussions, because we are just the biggest ribosome we've seen today is the human ribosome, and then people were just going off on tangents about why we're the most the highest, most dominant species because of that, and just people were biologists were getting in there and getting mad and everything. So.
It's not too surprising.
Yeah.
Do we have time for one more? Okay.
Cool.
So I've heard this idea before that you can see sort of the ribosomes of less complex species embedded in the ribosomes of more complex ones. But obviously, if you take humans and a fly, for example, those diverged in their evolution a really, really long time ago. Mm-hmm. So why would you necessarily find a fly's ribosome embedded in there? Do you think that there's intrinsic sort of evolutionary bottlenecks that only the ribosome can sort of get past, and it has to evolve particular features? Or is it just, you have to actually strip away some of these evolved features to find there's something on top of the fly ribosome that's not in the human one? Mm-hmm.
Well, I was looking at the structure, and so a lot of times you can have an AT base pair, or you can have ribosome, AU base pair or a GC base pair, and there's differences in that, but when you look at the structure of it, it looks the same approximately, but those sequences might be different, and there's some parts that are unique to different species. So, and plus that's just the RNA. What about the protein? Right. The proteins are very different between a lot of organisms. Some homology, but the same.
Got one more back here real quick.
Oh, okay, cool.
Have people done calorimetric measurements of ribozymes, measuring heat capacities and volumes?
Yeah, yeah, of course, I think so.
All right.
ITC experiments and everything like that.
Cool, thanks. I'm a thermodynamicist, so.
All right, excellent. Let's thank Nick again.